So the final talk is from Hector Baez. Hector is is a graduate student at the University of Rochester and is working with Dr. Gomez Ramirez. His talk is titled Microstimulation and Optogenetics, a combined stimulation strategy.
Great, thank you so much. So, yes, my talk is on a combined stimulation strategy, specifically to improve targeted activation of functional cortical ensembles using microstimulation and optogenetics. So, intercortical microstimulation has been used to study the functional role of brain areas and to provide sensory feedback in brain-computer interface applications. ICMS is most effective when applied to topographically organized encoding areas encoding similar features, such as in primary visual or V1. However, most feature representations in sensory systems, especially in somatosensory, are not topographically organized into columns, but instead are encoded by sparsely organized neurons. So using ICMS to target these motion ensembles may activate neighboring cells tuned for other features such as texture. An alternative approach is, of course, optogenetics, which enables us to activate or inactivate single cell at the single cell level. However, increasing intensity to cover a larger area loses the single cell resolution, and also we are increasing thermal toxicity. So the goal of this project is to reduce the ambiguity of artificial sensations by implementing a combined strategy of intracortical microstimulation and optogenetics using and to do this we're using a biologically constrained model of the SI cortex. So microstimulation and optogenetics each have distinct intensity distance relationships. Microstimulation is applied to cortical tissue through electrodes that deliver current spreading concentrically around the electrode tip. And the effective distance of excitation is proportional to the square root of the current intensity. On the left figure, neurons closest to the center of the stimulus are activated most strongly, of course, with the effect dropping off over distance. However, some neurons far from the center are still activated, likely from their axons traveling to the nearby source of stimulation or to local circuitry. And that can be as far as 1 millimeter distance from the center of the electrode. Luminous intensity originating from a beam of light, in contrast, dissipates rather quickly. And in brain slice, 1,000 milliwatts per millimeter squared in the center is sufficient to reliably activate a neuron, but this very quickly drops off. The model anatomy is anatomically organized based on the hand representation of the S1 cortex. According to Mountcastle's organization, each digit is assigned a spatial representation on our 2D plane, and neurons are spread out across 15 pad representations shown on the image, on the first two images there. The model is also comprised anatomically and functional interactions between excitatory and inhibitory cells representative of the EI balanced networks. Within those local circuits, we have a 1:4 excitatory inhibitory neuron ratio. And I have an example of this in the bottom left figure, where in this 2D representation, green dots represent motion encoding neurons, which we want to activate to induce some sort of motion percept. Blue are excitatory neurons encoding other features. Red are inhibitory, while black are axonal networks. And so we are using Poisson statistics to construct our spiking model. So the probability of spiking occurring in any time bin dt is simply r multiplied by dt. And within this simulation, we are modulating the r value. And so the way we decide if a neuron is spiking is if the neuron is spiking 50% of the time greater than normal, then we see that that neuron is activated. And our model also incorporates neural connectivity, where on the core scale, long-range projections can preferentially link cortical areas with similar responses to sensory stimuli. In this case, the similar responses encoding similar motion directions. So our combined neuromodulation approach is made possible through a transparent microelectrode array developed by our collaborators. And this array is capable of recording as well as delivering electrical current and also light stimulation to drive optogenetics. And so we used a particle swarm optimization to find the best stimulation patterns, which would selectively activate motion encoding cells. And we utilized this particle swarm algorithm to iteratively achieve these results, where a lower ratio represents a higher motion encoding selectivity. We found that microstimulation alone will always perform the worst, activating 50% more non-motion encoding cells compared to motion encoding cells. Optogenetics can be used in this setting as either an excitatory opsin, or in the purple in yellow as an excitatory opsin, or in the purple as an inhibitory opsin. And we found that the inhibitory opsin, as it currently is worked into our model, it works better than the excitatory opsin for creating a more robust artificial sensation. And yes, those are my conclusions. We were able to reproduce some in vivo data where we found that neurons closer to the source are activated much more easily, of course. But we were also able to determine that using this combined strategy, we're able to selectively activate functional sets encoding motion, encoding neurons. And we're also able to deliver these stimulation patterns with lower intensity profiles, which would result in lower toxicity for the cells. And that is all. Thank you.
Thank you, Hector, for the excellent talk. That was really interesting. So just feel free to post your questions on the Q&A, or you can also come to the stage to ask your questions. In the meantime, I can maybe ask, it might be a very naive question, of course. As I said, I mean, like, I'm really very unfamiliar with the technique. So do you think it would be possible, or maybe like as a next step, as a future step, it will be possible to use this technique and because there is efferent and reefferent feedback, to study the contribution of efferent and reefferent feedback. I guess in animals that's possible, but
I'd have to say that I'm not entirely sure. As long as using this technique, of course, we'll be able to study functional sets encoding very specific tuning, such as motion or texture, or if we were studying V1, of course, we could study orientation columns. So perhaps we could see something like this. Yes.
Intensity.
Perhaps.
So, like, if you think about the intensity of a somatosensory stimulus, maybe it might be possible.
Perhaps it's difficult for me to say for sure.
Okay. Okay.
Yes.
Yeah. I mean, again, as I said, I'm very unfamiliar with the method. So we have one other question from Devon Griggs. I hope I'm pronouncing the name correctly. Which opsins were used or would be used?
So in this case, we modeled two different opsins. One, we used a excitatory opsin and an inhibitory opsin. Specifically, they were quite forgetting the specific name of them, so I apologize on that. But functionally, the function of one is simply to silence the at the single cell level whatever cells were encoding non-motion, anything that wasn't encoding motion.
Okay, thank you. Also, thanks for the question, Devon. If you have any other questions, again, panelists or attendees, you can either come to the stage or post your question at the Q&A.
I have a question for Hector.
Sure.
So I missed that part, but sometimes when you excite a cortex with a current and you recruit recurrent inhibition, and then you have the paradoxical effect that you increase the current and then the cortex goes silent. So have you seen that, and are you always also going to model that, the recurrent inhibition?
So our plan currently is to model this in our model, but we are beginning this combined strategy in the mouse model right now. We are aware of that, but we haven't started yet, so I can't say for sure that we have seen it or not. But that is certainly a problem that we are aware of.
Thank you.
Thank you. Also, Gonzalo, for the question. So if there are no more questions, I think we can end our session. So I'd like to thank the speakers for the wonderful talks, and also attendees for their excellent questions and engagement. And also especially thanks to our backend, Rekha Varrier, who's been doing all the technical work. And with that, I wish you a nice day or evening or night.